The Ebola virus poses a significant threat to the immune system, targeting the very components of the immune system designed to combat viral infections. Upon infection, the virus immediately attacks the immune system, targeting the key defenders from viral infections: T cells, B cells, and natural killer cells. But how does Ebola virus do this? The virus achieves this suppression by initially infecting immune cells like macrophages and dendritic cells. Dendritic cells play a crucial role in transitioning from the innate immune response to activating the adaptive immune system, which fights viral infections. However, once infected, these dendritic cells lose their ability to present antigens on MHC2, which impairs the function of the dendritic cells in activating T cells. Remarkably, Ebola doesn't even need to infect T cells to kill them. Its surface glycoprotein triggers the death of nearby T cells, as well as natural killer and B cells, without direct infection. Even when antibodies are produced to fight the Ebola virus, the virus is one step ahead. The shed glycoprotein from the virus circulates in the blood, binding to antibodies and preventing them from targeting the virus effectively. Ebola has another immunosuppressive tactic, VP35, a protein coded for in the genes of the Ebola virus. This protein blocks type 1 interferons from being secreted by immune cells. Ordinarily, type 1 interferons turn on genes of the immune system to fight viral infections. The VP35 of the Ebola virus thus weakens the immune response. These numerous attacks on the immune system's ability to fight viral infections allows the virus to replicate at a remarkable speed. This ability of the Ebola virus to replicate quicker than most viruses after suppressing critical immune functions is a major reason for the lethality of Ebola virus. The rapid replication of the virus results in countless viral copies circulating through the body, each ready to infect new cells and invade vital organs like the liver, spleen, and even the central nervous system. As the virus spreads, the immune system ironically becomes a threat to the human body. The large amount of glycoprotein in circulation, due to the large volume of Ebola virus produced, leads to a tremendously large signal on immune cells to initiate inflammation. As this abnormally large amount of glycoprotein binds to immune cells, the body's natural inflammatory response spirals out of control. This results in more inflammation than the body is used to and, in some cases, capable of handling. This heightened inflammatory response, which is far beyond normal levels, leads to widespread damage across the human body. Part of this chaos involves fenestration of capillaries, ordinarily a normal part of inflammation, where cells create gaps to allow white blood cells to pass through. However, liver damage, which is common in Ebola virus infection, disrupts the production of blood clotting proteins, and kidney damage from the virus results in poorly filtered blood. This makes the blood more diluted and harder to clot. Consequently, with numerous fenestrated capillaries and increased blood volume, hemorrhage often results. This hemorrhage is a common cause of death from Ebola virus. What about deaths when there is no hemorrhage from the Ebola virus infection? Even if hemorrhage is avoided, the organ damage from Ebola virus is extensive. Ebola virus causes both a nearly unregulated viral replication rate and an aggressive immune response, which can lead to fatal outcomes such as encephalitis, cardiac arrhythmias, and multi-organ system failure. In essence, the Ebola virus's lethality results from a mixture of its ability to inflict widespread organ damage while simultaneously provoking an excessively forceful immune response marked by severe inflammation. Thanks for watching.